Okay, so today I'm happy to see half of the class are still here, not for fabric. But you will. Let's have a short class so you can all have a happy. Okay, so chapter 6 is about microbial genomics. Instead of just tell you what genomics is, I thought I'm actually lead you to actually analyze the bacterial genome and see how that feels. This is actually the first time I ever tried to do this in microbiology. I will see how it goes. If at the end everybody is still alive, I think I have accomplished something. So, okay, or everybody is still mentally. Okay. All right, so on Moodle, there's a file called Ecoli genome in R. Let's download that one. So, but make sure you have your RStudio already on your. Yeah, but for the HP, you need to re-download again because the Spellman HP, every time you download something, when you re-log in, it erases everything.
I think there's like the whole class here to go.
Yeah. No, the R is already there. You just need to re-download the RStudio. Okay. Make sure. Okay, let's ask. Oh, why don't we work as a team? If you don't want to work alone, I mean, Rina already have an Apple. She probably still have the RStudio from last time, right? Why don't we work as a team this time? Right. So. Nando, you want to join with?
I don't know if she's.
Oh.
But it doesn't matter.
Yeah. Tanya, you want to work with Makala in the back? She had the Apple laptop, probably already have the RStudio there.
Okay, I was waiting for Tisa, but
Oh,
she hasn't come in.
Okay, sure.
Who's Caleb?
Makala, why don't you move to the front and work
Oh, no,
with
she doesn't
Tanya?
have to. I'll move back here.
Oh. So, and Shelby already had the Apple, so so.
You can call me. You're talking about the Ecoli.
Yeah, download that Eco. Do you want me to dim the light, or you want to do it this way? Okay. Maybe if I dim the light, maybe you will see the screen. Yeah. So let's work as a team. Each team should have at least one computer with RStudio running, right? So at least able, it's going to run. So, and then go to Moodle. Let me see. There's a link Ecoli K2 genome in R. Click on that. They should download a zip file. Okay, so there is in my download folder. Well, if you are using Windows, you have to manually extract this. You have to, if it's a Windows, you want to right-click on the file, say extract all, and then the file will be extracted. Everybody clear? I see Bree are using Windows. JD, you are also using Windows. Make sure you download. After you download, right-click, extract all. Okay. So once we do that, and then you should open this. There's a one .r file. That's the R code. Say Ecoli K12.r. That's the R code we are going to use to analyze the Ecoli genome. I'm going to right-click on that. Say open with. And you see RStudio. So you want to make sure you use RStudio to open it. Sometimes it doesn't automatically open with RStudio with some other weird things. Make sure it's open with RStudio. All right. So this is what is everybody at this stage with me? Okay, then Yeah. Okay.
Is it .r or .st?
R.
No. So the Ethernet port into it.
If you are unable to connect to internet, make sure you use the internet cable. And the Ethernet cable. Oh, you already have it. You're opening with ARM, not ARM speed. Open with ARM speed. Excellent. I see. No, you have to download the network.
Yeah.
Okay. Yeah, you have not saved it. Oh, you have to install. After you install, it should be in. Oh, maybe you haven't installed. And download make sure the file is downloaded.
Oh, I'm like, where the hell, bro? I'm gonna try to do so about 7 o'clock. He is already done with the social media. Your exam starts at 5. Well, we're trying to get it done. Let's do it now. So we're gonna. You know, oh wait. Yeah, of course. You do from 5 to 7. No. Actually, we're going to 4.
Okay. Once we are open this R code on, yes. Joanna, can you join Carrie? Yeah. 2 people working together will be much easier.
I have a quick question.
Join Michaela on the other side. Yeah.
Do we have to do the source allocation?
Excellent. Yes. So yeah, we need to do that. So once we want to make sure we are working at the right directory, go to a session, set working directory to the source file location. Okay. So excellent. Some of you actually remember how to do this. Once we done that, we need to install a tiny little package. It's actually almost like an app on your iPhone or your smartphone, other smartphone. We need something called sequence input and SEQINR. So we need that one to install on your RStudio. So you just highlight that and click run. It should install the hopefully it takes just a few minutes to install it. Okay, on my computer it's very fast. I just finished it. Said the error?
Yeah.
Ooh. What error is
What was it? There's no package.
Oh my goodness. I need to copy paste the original one. Okay, I guess why is this? Okay, let me. I guess I have to copy paste the actual code. BioC. Right. G. Okay. Okay, I'm going to change it to a different one. Hopefully this time it works. I'm going to save this file as Ecoli version 2, and then upload to Moodle. Yeah, unfortunately, on my computer, it looks like it works a little different from your computer. Okay, remove the old one. Okay, I'm going to go back to the Moodle and just delete the old one. And then put a new one there. Okay, okay everyone. I just updated the new code, the zip file, the R script on the Moodle again. So if you download again, let's see, the code should change. Hopefully this two, that single line now replaced by two different lines. Hopefully this time it will work. Yeah, I replaced that line with a different code, different lines now. And once you download that, you again need to set the working directory to source file again. And then you can highlight that. I'm going to wait until everybody on the same page again. Yeah, download the new one. I removed that one, so I changed that one to something else. Let's see whether this time it works. Yeah.
Maybe.
So you can download it again. So. Okay, so you see that I change the working directory again to the source file. Go to a section, and then run that two lines. Okay. You don't see that. Okay, yeah. run the first line, see what happens. First two lines. What is it? Yeah, say update all. It asks you, do you want to update all? Say A. It's going to ask you, do you want to update all, some or none? Choose all.
They're gonna ask you all none or some. Just put A to answer.
If you look at the bottom, it says, do you want to update all? No, you have to run the first two lines and then you choose say update all. Yeah.
The first one.
Just type A. A is for between A. At the bottom, you type at the top. You type at the bottom, remove that A. Remove that A. You just remove the two lines. You have to remove the entire two lines.
So you just.
Why not?
Don't press open. I know we go to open file and click on this file.
So make sure you are connected to the right window.
You could say it.
At the bottom left, select the bottom left, click all. Say the bottom left, click all. So once it finished,
So do we type for the Windows people? Do we type A and then press enter?
Yeah.
I was just thinking it's time.
yeah.
I did it today. Didn't know. So what were you asking?
You know those parts he said.
Well, do that.
Yeah, it was confusing.
Oh, but it is.
Yeah, then it's done. Once it's done, so some of you already finished installation. And then move to line 3, say require that package. This, and at the bottom you see loading that package into our software. And then we can run line 6. This is just to make sure we are in the right place. You should see two files. One is the R code itself. The other one is the Ecoli K12 genome. That's the Ecoli strain K12 genome.
So we should run 6.
Yeah, run line 6. You should see. Yeah. Once you can do that, run line 8. This will be load the Ecoli genome into RStudio. It's take a few seconds. Yeah. You say the error?
I don't know. I think our line 8.
So go to line 8 and then run it. Is that what you did?
Yeah.
Yeah.
You haven't changed your file directory. You are still at the subdocument. Change the file directory to this file. Go to a session, change file directory. Okay. So make sure you are in the right directory. I still see some of you working at a different directory.
So you change the directory.
Actually, you can see the Ecoli genome file in the source file. Make sure you have run every line. Some of you didn't even run the first 3 lines and then just go directly into number 8, try to run. You run it, follow me, run the line by line. Don't jump over it.
I thought we read. So you run 1 and 2, you run 6, and you run 8.
Right. Yeah.
Yeah, I still got it.
Okay. So by now you should have the E. coli genome read into the sequences. Sorry, what? What error do you see? Have you run line number 3?
Oh, no, it's supposed to be like
So you want to run on 3, 2.
You want to run first? You run number 9, 1 and 2, then 3, then 6, then 8.
I missed.
No, I was just ignoring. She's like, here today to look at it. And I was like, oh, I checked it today. And I was like, oh, this one, actually, yeah, you can have it. There's so many. Have you been there? Real drama.
Y'all might need to read the week. I didn't see that. Oh, yeah, mine is like.
I'm gonna try it again. You publish on 3 times. Oh, so that's too many.
I don't even mind.
That drinking from the side.
No, this side right here. So it should do it.
Okay, now line number
1 is the first 2, 3, 6.
I'm reading 6.
no, you should
Alright,
finish.
No, it's okay. So we're good. Oh, okay.
Then go back from
So this was line 3, then 6.
you have to change your working directory to the next one, change the working directory to
This one. Come on, Mom.
Okay, so once you run the first 8 lines, and you will see the E. coli genome will be in has been read into the R Studio, and it's actually I call it the sequence. It's actually on the right. It's this 5287 element. Basically, there are 5287 genes in the E. coli genome. Right. So if I click, and you actually can see the first one. Oops, let me see if I move it a little. The first one is it's called NT01EC E. coli. NT basically is the NCBI accession header. This is the EC for E. coli 0001. That's the first gene annotated. And this is some format called FASTA. And it's on chromosome. E. coli have just 1 chromosome. So this NTEC 001. And there's actually there's actually a long name. If you here, if I put my mouse here long enough, you can actually see this is a gene called bifunctional aspartokinase homoserine dehydrogen something. So that's the first gene. So if you put your mouse longer enough, you think it seems to pop up. Oh, why is it stop? Yeah, here. That's the first gene. And if you move to the second one, you see the second gene. This is the homoserine kinase. And if you scroll down, you can see many other genes. That's this one. This one is the DNAK protein. Well, they are more than 5,000 genes. So E. coli have more than 5,000 genes in this one. So this one, this is the hydrogen dipole kinase. I don't know what this gene. I've never heard of this. Anyhow, there are 5,000, more than 5,000 genes, quite a lot of them. You can actually run line. So this is on number on your right hand side in the window. On the left hand side, if you highlight the line number 10, just run that. It actually gives the same thing, but it's just output in the bottom left window. This function called str is said to look at the structure of the sequences. This seqs sequences. It's just a short name I use. So that basically is E. coli genes. There are 5287 E. coli coding genes there. Now, how do we look at the first gene? You can do that. Say sequence 1 is that, and then you look at the top. Sequence 1 is there. How do I get the name of sequence 1? Just say get name sequence 1 exactly shows here. Right. NT01EC001. So instead of looking at the right, I can do this one. And then how do I know? Here's it. So the sequence is out there. So then you can do some explorative things, investigation, right? And so this is a gene. And if I ask you how many A's, how many G's, how many T's, how many C's in this gene, how do you do that? And we can use a command called table. Then it actually shows at the bottom. So, maybe I should put this a little higher so you can see this.
So highlight the table and run it.
Yeah. Highlight table and click run. You will see here. So how many A's it has? There are 553 A's, 615 C's, 692 G's, and 603 T's. This is in the sensing direction.
You have to highlight 13 again. Run it before you 15.
Yeah, don't just jump over. You want to make sure you run the previous line. If you just run the last line, nothing will happen. So, yeah. Okay, so this is basically nucleotide composition. Okay, given this data, if I ask you what's the GC percentage in this gene, I'm going to ask you. Sorry, what? How do?
GC, the amount of GC.
Yeah, what's the percentage of GC in this gene? How do you do that?
Add all of them, and add GC.
Yeah, why don't you? How do? Okay, so basically you can do this. 615 plus 692, right? And then divided by 553, add 615, add 692, add 603. Well, that would be 53%. Well, here in R, if you highlight the line 16, let's see whether you get the same thing. Well, 0.53. Right? So that command GC just calculate the GC percentage for the gene.
Dr. Chen, so I just run from 13 to 16 and give me the answer.
Yeah, sure.
Oh, okay.
Yeah, if you highlight the entire code, you're going to run the whole thing over.
Yeah.
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 Right, yeah. So, all right. Now I'm going to ask you do your own exercise. Why don't you get the gene name of get gene name of 789 gene? How are you going to do that? Yeah. Instead of get the first gene, I ask you to find out the gene name for the 709 and the GC content. GC content of that. Of 789 gene. I'm actually curious to see how many of you can get this. Right, I told you here, this is the first gene. But if I want to look at the gene, say 789th gene.
So you put 789.
Yeah. Yeah, it should just work. Yeah. Right, and let's work on it for a few minutes. See how many of you can get the answer of this.
So what are we trying to do?
GC content of the 789th gene. What's the how many?
It's NT01EC0800.
Okay. What's the percentage of GC content?
Can you show how you do that?
GC.
You got a 50? Well, so let me also do this. So I can change, basically remove that 1 to 789. So 789. And then run this code again. Let's see. Yeah, very good. 50%, yeah. So, and the name is the ECOA00. Yeah. So. Yeah. So.
Did you type that line 17 in there?
Yeah, I typed the line 17 there to basically tell you what to do. Yeah, get the gene name of 789 gene. Sorry, what?
If I type it in, it should give me a name.
No, it doesn't. That's the question. Yeah, that's a comment. Yeah, so that's just I write something to ask you. That line doesn't do anything.
Randy, you see you're supposed to add
Sure.
you gotta get an area.
Go back to the first one where it says sequence 1. Highlight that, run it, and then go to the table. Should we type in number 17? Question you asked.
No. You can. That's I can just remove that. Yeah. So that's a comment.
So you run from 13 to 16.
Yeah, I just run from 13 to 16 and give that. Yeah. So I can remove the line 17. I wrote there just to make sure clear you know what the question is. So, yeah. Okay, so now what if I ask you, say, look for gene 89, gene 102, gene 504. I give you a whole list of genes. That to do it one by one doesn't seem to be reasonable, right? It's pretty tedious. So this is the whole purpose of why we have to do programming. We can ask computer to do this automatically, right? So there's a one technique in some of you, if you have some of you have taught computer language, you can do a loop, right? It's very boring for human to do the same thing again and again, right? So, but it's actually computer just does this automatically. If you ask computer to do the same thing again and again, it's actually very easy and very accurate. If you ask a human to do this again and again 1,000 times, it's probably guaranteed to make some mistakes somewhere. So, okay, so this is how we let the computer to do this. So there are more than 5,000 genes. I'm going to ask the computer to calculate the GC content for all of them. Instead of doing it one by one, I'm going to ask them to calculate the GC, get the name and GC content for all of them, then output it. So, and this is basically, if you highlight from line 19 to line 27, that's what this is going to do. We just run that. It's going to, see, actually it doesn't take very long. On my computer, it just pop out right again.
So
Yeah.
And basically, every result in the table called out. I'm just showing the first 6 lines. So the first row is called GC content. The second column is gene name. And if you don't like that, you can go to the right-hand side. You see there's the out table. You click that, there. Actually pop up this is basically like a spreadsheet. There's a table there, right? There's that's the number 1 GC content, gene name, percentage. That's basically GC content. They should be more than 5,000. So this will be a very long table, right? So let's see. Yeah, 5,287. Right. So there, and we just calculate the GC content for every gene in the E. coli gene. So, and if you recall, in the textbook, it actually shows E. coli genome there how many genes. Actually, that's really not the case. If you look at the different strain, you will get different numbers. So there's really a range of the E. coli genome. The bacterial genome is much more fluid than the human. In human, there are also some variability. That's called copy number variation. Some people may have genes say have more than 1 copy than other people. That's actually how different people why everybody is one of the reasons why everybody is different from others. Okay. Yes, great.
In this coding, why did it only do 1 through 6 if there's more than 7?
No, I just say header. This header command just show the numbers first to 6.
Oh, okay.
Yeah, that's just to look at the number 6. But in reality, that out one stored more than 5,000 rows of data. Yeah, but if I want to show 5,000 rows in one screen, I can't even see anything.
So if you want to show 1 through 10, would you put 10 in the out? Or?
Well, if you do that, you just say out, say 1 to 10. That's just the 1 to 10. Right, so if you want to see, say, file 1 row and 103 row, 999 row, that's just.
So we should know the command. Should we know the command?
No, you don't have to know. You just this is not programming class, but I'm just doing this to give you exposure what computational genomics is. What's the people at the CDC or I don't know if let's say there's a new infection disease, a bacteria just being discovered, we sequence, how do we do follow up, right? So okay, so this is basically almost like just let you have a sense what this thing is, computation. Why do we do this, right? So I'm not requiring you to learn how to do this. Yeah. So. Why is that a relief? I don't know. But anyhow, but after computer, you can also save the data. Let's say your boss say, well, how many genes is this? You don't. Oh, this is this. But you have to write a report and put the supplementary information. So we can output it. So if line 29, you basically write this file to your computer. So if we run line 29. After that, if you go back to your file folder, you should see a file on your own computer called gc.csv. And if you double click that, that should open up in Excel. So I'm double clicking that file, that should be open up in my Excel. Hopefully my Excel is running. Yeah, it is running, it's just a little slower. Well, I guess it's really slow. Okay, finally. So, come on.
This is cool.
Yeah, this is cool.
Oh, sure. Thank you. At least someone found it interesting. That's yeah. So that's basically the whole thing, right? So, and if I scroll all the way down, I should find how many how many rows? It'll be 5287. Wow, 5287, I guess is really a long list.
To 5,000?
5,287. Oh, well, 5,288 because the first row is my column name, right? So there. So, okay. All right. So there. So we have just generated an Excel report for the calculation we did, right? So you can send your boss. Hopefully he or her is impressed. So, okay. Anyhow, now we can do some other analysis. We can find out how long each gene is, right? So this is, if you highlight line 32 and 33, that's basically to calculate how long each gene is. I basically found the first nucleotide to the last nucleotide. So if you run that, and this time, oh, it's also very fast. And we can line 41 are going to be output again to Excel file. So you can look at this file on the right-hand side. That's number 2, right? Gene name, gene length, plus GC content on the right? So this time we do more calculation. This time I calculate gene length and the GC content. But if you go to your computer file folder, you should see a second file now. GC number 2.csv. You double click on that. That again should be a larger. Yeah, that's there. Gene length, GC content. This time you have gene length there now. So, okay. If we go back to the R code, now this time I'm doing something different, right? So if you look at nucleotide sequence, usually it doesn't make too much sense for protein function, but we can translate those into protein. If you recall in some other courses, and your professor may give you a DNA gene and ask you what's a protein of it, what's this gene is, you have to translate it into protein. And here we can also ask computer to do this automatically. So if I want to translate the first gene, I just type translate. That's actually is a command built into this. I can type translate protein, but once I translate, I don't call it a gene anymore. I call it a protein, right? Just to make a difference. So at least we won't be confused by the things we are working with. So highlight that. There. On the right-hand side, you can actually see a protein show up. And how do we find out the length of the protein? You can highlight number 40, line 44, and you will see 882. That's the length of the protein. And if you just type that protein directly, you can see the nucleotide, not nucleotide, sorry, amino acid residue in this protein. So there. So first one is methionine. The last one is a star for stop codon. So this is protein coding gene just been translated. So, in fact, I can also remember how I calculate how many nucleotides using the table function. I can do that also for protein. If I tabulate every residue again, you see at the bottom now, I can see, wow, there's 63 glycine in this protein. 20 Y. What is Y, everybody? What is Y? I think it's tryptophan. No? Is it tyrosine?
Wait, let me check.
What is a K? K, I think, is a lysine. Yeah, but you can check. A should be alanine. C should be cysteine. D, what is D? I always have trouble with D and E. What is D and E? Why don't you Google? So, what is D and E amino acid? H must be histidine, I will be isoleucine, L will be leucine, M is methionine, N I'm having trouble. P should be proline, Q I'm having trouble again. R, maybe R is arginine, S is serine, T is either tyrosine or tryptophan. We should be validating W. W is tryptophan, I think. What is Y? Y is lysine? Yeah, maybe you're right. Yeah, I don't remember. But when I was in college, I remember every one of them. So, what?
It's tyrosine.
Tyrosine? Oh, thank you. All right. Okay, so, but again, this will be okay. Now let's do another exercise. Why don't we translate? Let's translate the gene number 999. How about this?
You said 900.
Translate the gene of 999 and tabulate the amino acid in that protein 999. Oh, first tell me how long is that protein is, right? So why you say that? Okay, let me see. So I'm gonna also do it here. I basically change the sequence to 999 and then see the length. Oh, 139. Oh, excellent. And then we tabulate the. Well, this is a very short protein, so yeah. So it's of course the amino acid, the residue will be also fewer than the previous one. So yeah. Okay, so now the next one is I'm going to this time I'm going to do it again. Instead of doing the one by one, I'm going to translate every protein coding gene in this Ecoli genome, and then take the name and also calculate the length of the protein here. So if and then I'm going to output to a file. This time I call it gcprotein.csv. So I'm going to highlight this whole 49 to 59, this 10 lines of code, and just run it. Oh, it's done. So if I go back to my file again, so there's this time there's a new Excel file just being output. Let's open that one in Excel. There, that's the right. So the first gene name, open reading frame length. That means the gene length, and then the protein length. What's the relationship of these two again? From gene length to protein length, it should be what? It should be shrunk by 3 times, right? So each protein residue should be correspond to 3 nucleotides. So if I do this, say OF length divided by protein length, that should be 3, right? So that if I in Excel, if I do this for every row, they should be all 3. If it's not 3, then we are having a problem. So let's just for the heck of exercise. Oh, they are all 3. So yeah. So you can double check whether this genome is correct or not. But every now and then you will see actually that's not the case. This is because in some of the bacteria or mutant, apparently they can correct some tiny mistake. In fact, I remember in one of the mutant genome, there is one gene has an extra nucleotide, but the protein is correct. That's because in some peculiar case, the bacteria can somehow correct that single nucleotide mistake and still move on to it. So biology is a wonderful thing. You think it's always should be 3 times different, but every now and then there will be an exception to that. So, okay, but sorry. Okay, so let's see. Yeah, that's it. That's the end of the exercise. I mean, let me stop the video.